Assalamu alaikum wa rahmatullah. I'm Rawan Twaji, and today I will introduce you to the molecular basis of carcinogenesis. In this video, I will talk about a principle relating to cancer. I will also highlight the hallmark of cancer with a specific focusing on a proto-oncogene, tumor suppressor gene, DNA repair gene, and gene that regulate apoptosis. So let's start. Bismillah. So what is cancer? When you have uncontrolled division of the cells that will give you a mass which is malignant, this is known as a cancer. Cancer caused by a mutation in the DNA. However, not all the mutation will cause us cancer. There are different types of a mutation. First, lethal mutation, which will cause a cell death. So when the cell dies, there will be no further division and there will be no cancer. Second type of mutation is a neutral mutation, and it means that the cell is still normal even when there is a mutation. So further division of the cell will give us a normal cells. Third type of a mutation is an alteration mutation, which will cause the cell to have a mutation in the DNA, and further division of the cell will give us a daughter cells with the same mutation, and this is the cause of cancer. There is a principle relating to cancer, and it's carcinogenesis. So carcinogenesis means non-lethal mutation. So when we have a non-lethal mutation, this will give us cancerous cell. Cancerous cell also is monoclonal in origin, and this means a one mother cell will give us the whole mass of cancer. So this means that the whole cells in this mass have the same genetic information of the first mother cell. So what is the character of cancer or the hallmark of cancer? Cancerous cells should be self-sufficient in growth signals. It should be also insensitive to anti-growth signals. It should also have the ability to invade and metastasize. It should have also a limitless replicative potential, the ability to have its own blood vessels by angiogenesis. Also, it has a defect in DNA repair and evading the apoptosis. So normally, the cell in your body has to go through division. This is in case of personal growth or if there is an injury and we need a replacement. So the gene that driven the cell to go through this division called proto-oncogene. So proto-oncogene promotes cell proliferation when there is a need. So when we need a replacement and we need a further division and the body is in need, we will have activation of this proto-oncogene and we will have a further division. However, if this gene gets exposed to radiation, virus, chemicals, or it inherited with a defect, this gene will be mutated. And when it's mutated, it will acquire a new name and it will be called oncogene. So oncogene is a cell proliferation when there is no need. So when there is no need for cell division and there is a further cell division, this is caused by an oncogene. So this will lead us to have the first criteria of the cancerous cell, which is a self-sufficiency in a growth signal. So this means even when you don't need a further division, even when you don't have a signal, you will have a growth. So proto-oncogene is a group of genes that promote the cell growth. Every type of these genes will express itself into a different type of a protein. So first gene will give us a growth factor receptor, and the other one will give us the growth factor itself. The third one will give us the transducer. The fourth will give us transcription factor. Also, we will have a gene for cyclin and a CDK, or which called cyclin-dependent kinase. These all will act together in a harmony to promote cell growth. So to understand what could go wrong with the case of oncogene, we should understand the normal action of a proto-oncogene inside the cell. So after highlighting that, we will discuss the oncogene and what could go wrong. First of all, we will speak about the proto-oncogene. So proto-oncogene aim to click the DNA synthesis arrangement, and this is by activation of E2F. So there is a different expression of this proto-oncogene that's aimed at the end to activate E2F. So as we highlighted in the previous slide, we have the growth factor and we have the growth factor receptor, we have the transducer, we have the transcriptor, we have the cyclin, and we have the cyclin dependent kinase. So how will these act together? So first, the growth factor will bind to the growth factor receptor. So the growth factor receptor will be activated. After that, the growth factor receptor will activate the transducer. So the transducer will acquire the GTP. So when the RAS get activated, it will send a signal to the responder gene. The responder gene will be activated by producing a transcriptor factor. This transcriptor factor will go to the next gene and it will activate it to produce the cyclin. So the cyclin will act with the cyclin dependent kinase. So they will come together and they will go to the protein that caused the deactivation of E2F. They will go to this protein and phosphorylate it. When this protein become phosphorylated, it will be deactivated. So the E2F will be activated and the DNA synthesis will start. So now I just wanted to highlight important names of these components. So one of the most important transducer called RAS, and one of the most important responder gene called MEK gene. So this is what could the action of a proto-oncogene be. The proto-oncogene also could act through ABL, and ABL is a receptor that does not need any growth factor, and it will act directly to activate the cyclin gene to produce cyclin, and then the further steps will go. The cyclin will act with the cyclin dependent kinase to phosphorylate the protein that cover the E2F. It's important also to mention that the protein that cover the E2F called a retinoblastoma or RP. So this is the normal. What could happen in case of oncogene that will lead to uncontrolled cell division? First, we might have a very sticky growth factor that when it's bind to the growth factor receptor, it will bind forever and it will cause a forever activation of the growth factor receptor. Also, growth factor receptor might be activated without even a growth factor binding. Also, we might have a lot of a growth factor, or we might have a lot of a growth factor receptor, so we will have a more than normal division. Also, in case of a transducer, this could be a site of defect in case of a proto-oncogene. So normally, when the RAS protein become activated, it will activate it by the binding of GTP, and then after sending the signal, it will be deactivated by the action of intrinsic GTPase. So in oncogene, we will have a lack of this substance, so the RAS protein will be activated forever. Also, we could have an overactivity of MEK gene. So we will have a lot of transcriptor, so we will have a lot of cyclin and then a lot of activation. Also, the abnormality could be associated with the ABL. So when we have a further activation of ABL or unneeded activation of ABL, so this will cause us to have more cyclin, and then the process will go on. Oncogene is a mutated picture of a proto-oncogene because it has a more than normal action of a proto-oncogene. So the type of a mutation here is a gain of function mutation. And this gain of function mutation could be expressed in different type of mutation. So for example, we will have the point mutation, and this means we have an overfunction of the different component of the proto-oncogene. So in case of very sticky growth factor, or the growth factor receptor that act without even activation from the growth factor. And then also the type of a mutation could be a gene amplification. So for example, you might have a growth factor receptor, only one gene that express the growth factor receptor in the cell. So when this gene become amplified to more than the normal number, we will have more than normal number growth factor receptor. So we will have a more than normal number division. The third type of mutation is called translocation, and this means sometimes we have a proto-oncogene as in the specific chromosome. So the gene here is affected by the activity of the next genes. So it will be adapted to certain limit of the activity. So when it translocated to other chromosome and become next to a very active gene, it will be overexpressed. When it become overexpressed, we will have a more than normal cell division, and the proto-oncogene will be converted to oncogene. Also, an important fact that you should know, that the gene has two alleles, one from the mother and the other from the father. So in case of a proto-oncogene, if the one of this allele become mutated, it's enough to express the defects here. It means that when you have one defect, for example, let's say that you inherited a defected proto-oncogene or oncogene from your father, so you will have further or abnormal cell division. So this means that the inheritance pattern of oncogene is an autosomal dominant pattern. So here I just wanted to highlight the different types of proto-oncogene mutation and its clinical correlates. So the first type of a proto-oncogene mutation, and which is a high yield fact, is the mutation in RAS. So a mutation in RAS is the commonest proto-oncogene mutation, and you should know this by heart. So if we have a mutation in RAS, we will have a colon carcinoma as a condition. Also, when we have an ABL mutation, we will have a chronic myeloid leukemia. Also, we could have a mutation at a MYC gene, and there is a different type of MYC gene, like c-MYC, which is correlate with Burkitt's lymphoma as a condition. L-MYC, which is correlate with a lung cancer, just link here the L with the L. N-MYC, which is correlate with the neuroblastoma, and link here the N with the N. So even when you have an oncogene in your cell, this is not enough cause to develop a cancer. This because you have a tumor suppressor gene in your cell, and this gene acts to inhibit a cell proliferation. So it acts as a car brake. So it will tell your cell to stop further division. Unfortunately, this type of gene is mutated in case of cancer, so the cell will continue to proliferate and it will not respond to this kind of gene. So in normal people, when you have a more than normal division, the tumor suppressor gene will act to stop this division. Unfortunately, this is not available in cancerous cell. So this will lead us to have the second character of cancer, which is insensitivity to growth inhibitory signals. There are many types of a tumor suppressor gene. However, in this video, I will introduce you to the three most common tumor suppressor genes. First, ABC gene. Second, P53 gene. And third, RB or retinoblastoma gene. So do you remember this picture from proto-oncogene? With the same picture, we will explain the different types of a tumor suppressor gene. And we will start with the ABC gene. We will discuss the normal action of ABC gene, and then we will discuss what could go wrong. So before talking about the ABC gene, we should understand the action of B-catenin. So B-catenin is a type of a protein that it will goes to the responder gene or MYC gene, and it will cause the transcriptor factor to be produced. Then, as we know and discussed before, it will transcriptor factor will cause us to have the cyclin, and the cyclin then will act with cyclin dependent kinase to phosphorylate the RB, and the cell division will happen. In case of the action of the ABC gene will inhibit the action of B-catenin. So it will inhibit the activation of MYC gene, and then there will be no cyclin, and then it will not combine with cyclin-dependent kinase, and phosphorylation will not happen, and the E2F will remain inactive. However, if we don't have the action of ABC gene, the B-catenin will be active all the time, and then we will have the cell division goes on and on. Then the second type of a tumor suppressor gene, and it's the most common site of a mutation in all cancer, is P53. So P53 will act directly on the cyclin-dependent kinase, and it will inhibit the action of cyclin-dependent kinase by cyclin-dependent kinase inhibitor. So there will be no phosphorylation of the RB. However, in case of cancer, we will have a mutation for the P53. So the cyclin-dependent kinase will act and phosphorylate the RB. The third tumor suppressor gene is the RB. And the RB will act as a cover or inactivate the E2F. So it will cover it, and it will inactivate it and keep it in inactive state, so to prevent any cell division. However, in case of defects in the RB, we will not have a cover, or we will not have an inactive state for E2F. This means that the E2F will act all the time. Even the proto-oncogene action in case of RB defect is not needed, or the oncogene defect is not needed, since the action of oncogene and the proto-oncogene is to phosphorylate the RB to expose the E2F and cause cell division. So it will be activated all the time in case of defect in the RB. Tumor suppressor gene mutation is when the gene lost its ability to prevent cell proliferation. So the type of a mutation here is loss of function mutation. And this could be by two types of a mutation. One is point mutation, and this means that the gene and the gene protein is presented in the cell, but they are not acting well. The second type of a mutation is deletion, and this means that there is no tumor suppressor gene or tumor suppressor gene protein in the cell. And the deletion could be two types, interstitial or chromosomal. Interstitial mean when you lost a part or a segment of your chromosome, and chromosomal mean when you lost the whole chromosome. Tumor suppressor gene is autosomal recessive, and this means you need two alleles to be defected to have a tumor suppressor gene mutation. So when you have inherited from your father a defected tumor suppressor gene, you will have the other alleles from your mother working well. And if there is another hit by the environment, the somatic hit or the somatic mutation, then the tumor suppressor gene will be affected here. And you might have the both allele hits from the environment. So you have the hit for your father alleles from the environment, and the hit that affects the mother allele also from the environment. So you are required to have two defected alleles to have a tumor suppressor gene mutation. So now let's see different types of tumor suppressor genes mutation and their clinical correlations. So first of all, we have ABC mutation. ABC mutation is known to be linked to FAP or familial adenomatous polyposis and colorectal cancer. P53, on the other hand, is the most common type of a mutation among all the cancer. Don't confuse that with RAS. RAS is the most common type of a mutation in proto-oncogene, and also P53 known to be linked with Li-Fraumeni syndrome. The other type of a mutation is the RB mutation, and it's known to be linked to retinoblastoma and osteosarcoma. Even if the cell has an oncogene and a tumor suppressor gene, it still has a chance to not become a cancerous cell. This because it has a DNA repair gene, which concern about correction of the damaged DNA. So unfortunately, this type of gene is mutated in cancer. So there will be a DNA damage and instability. So this will lead us to the third hallmark of cancer, which is known as impaired DNA repair mechanism. There are different types of DNA repair system. We have mismatch repair system, nucleotide excision repair system, homologous recombination system. So mismatch repair system, when you have like here thymine linked to guanine, and this is a defect, the thymine should be linked to adenine. So this type of system, which is the mismatch repair system, will correct that. Sometimes you will have a defect in this system, and this is linked to condition known as a hereditary non-polyposis colon cancer. The second type of system known as a nucleotide excision repair system, and this when you have a defect in the sequence of a nucleotide. So the whole sequence of a nucleotide is defected. So you have to take this out and repair it. And if there is any defect in this system, this will give us a condition known as xeroderma pigmentosa. The third type of DNA repair system known as a homologous recombination system, and this repair system will work when you have a break in the DNA. So your DNA break into half. So BRCA1 and 2 will come into action, and they will link these two together. So always remember, and this is a very important information, that BRCA1 and 2 are DNA repair genes that are concerning with the homologous recombination system. The type of defects here is breast and ovarian cancer. So when you have a defect in BRCA1 or 2, the breast and ovarian cancer will be the condition. So if the DNA damage is so severe to the extent that the DNA repair gene cannot repair it, or if the DNA repair gene is defected, the apoptosis is still there. So the cell could go through the programmed cell death if the DNA damage beyond repair. Unfortunately, this type of ability is mutated in cancer. So the cancerous cell has no ability to go through apoptosis. So the cell lives for a long time. So this will lead us to have the fourth hallmark of cancer, which is evading apoptosis. So for apoptosis to happen, caspase pathway should be activated. So for that, we have different types of gene regarding apoptosis. Ones which will prevent the caspase, so it will promote the cell life, and the other will activate the caspase pathway, and it will promote cell death. So anti-apoptotic genes, which is mean that it's against cell apoptosis, like BCL2 gene, it will prevent the caspase pathway. So it will prevent the cell going through apoptosis and promote the long life of cell. The other type of gene, which is called the pro-apoptosis gene, this gene supports the cell death or the cell suicide, and it will activate the caspase pathway. One of these genes known as BAX. So in cancerous cell, we will have an increase in anti-apoptotic gene and decrease in pro-apoptotic gene. BCL2 gene is anti-apoptotic gene, as we mentioned, and the mutation of this gene linked to condition known as follicular undifferentiated lymphoma. So let's practice that now. If we want cell to live longer, we will increase the BCL2 because it's anti-apoptosis and decrease the BAX. And when we want the apoptosis to happen, we will decrease the BCL2 and we will increase the BAX. Also, cancerous cell has unlimited replication capacity. So normally, the DNA has telomere in its both ends. This telomere will get shorter and shorter through cell division. Till the time it finishes, then the cell cannot go through further division. However, cancerous cell has developed a way to keep this telomere long. This is by telomerase. So the cancerous cell has the ability to go through unlimited replication. Also, cancerous cell has the ability to build its own blood vessels. This is through a process called angiogenesis, and this is through producing a substance called vascular endothelial growth factor, or VEGF. Also, cancerous cell has the ability to invade and metastasize to other organ and tissue. So, after discussing all these 7 hallmarks of cancer, now you have a malignant cell and the cancer has developed. So, these are the references for this video. Thank you for watching. If you have any comments or suggestion, communicate us through the email. Have a nice day.